Welcome to the Leaving Cert Biology channel. This ties hand in hand with our website leavingcertbiology.ie, which is a platform for 5th and 6th year students in Ireland studying Leaving Cert Biology. Of course, we have our TikTok and Instagram page at leavingcertbiology.ie as well, where we offer completely free videos, tips, tricks, exam strategy and trends. And on our website, we have exam paper structure, timing, all of our completely free podcast episodes, and so much more. Our aim is to support students all over Ireland. So in this episode, we are following on from last week and we are doing the highly requested genetic crosses part 2 dihybrid cross, which is highly anticipated to come up for 2025. I would strongly, if you haven't already, advise going back over last week's video and looking through all of the definitions on the genetic crosses and the theory that you can be asked before you actually look at me doing the examples on this. Last week I covered the first one there, monohybrid, and today I'm going to do dihybrid and incomplete dominance. If you'd like to see the remaining two, comment below or please let me know. Okay, so let's get started. Here I have an example, so let's read through it and show you how I approach a dihybrid question. So dihybrid, remember, is crossing two traits. You must have two alleles for each trait. They can either be the same, homozygous, or different, heterozygous. So let's read this question. In a species of plant, yellow flower, capital Y, is dominant to green, small y. That means green is recessive, yellow is dominant. If I have homozygous dominant, I have two capital Ys, homozygous recessive, two small y's, or heterozygous, one big Y, one small y, and the phenotype would be yellow because the dominant is always expressed if it's present, except for incomplete dominance, which we'll see in a minute. Now my second trait, I have round, capital R, is dominant to wrinkled, small r in shape. So wrinkled would be recessive, dominant is round. Then we are asked to cross a yellow round seed that is homozygous dominant for both traits with a green wrinkled seed. Green is recessive, wrinkled is recessive. The only way that a recessive can be expressed is if I have two of them. So let capital Y be your yellow for dominant, your small y is green for recessive, your big R is round, which is dominant, and your small r is wrinkled, which is recessive. So we are asked to cross a yellow round seed with a green wrinkled seed. So let's do our steps. Always start off with your parental phenotypes, what they physically look like. pH for phenotype, pH for physical. So homozygous dominant means homozygous. The letters are identical. So we either have two big Y's, two big R's, two small Y's, two small R's, and the dominant tells us whether we have a capital or a small. So homozygous means we have two of the same alleles, and the dominant refers to that they are both capital. Okay, so you will see here my yellow round is parent 1 and my green wrinkled will be parent 2. The genotype is what I have to do next. It's homozygous dominant, meaning dominant is capital Y, homozygous I have two capital Y's, round is capital R, I have dominant homozygous, so I have two capital R's, two capital Y's. Remember, you must have two alleles for each trait. Green wrinkled then is two small Y's and two small R's because that is homozygous recessive. The next thing we have to do is look at our gametes. Remember, each parent can only pass on one allele for each trait. So the only combination of gametes that the first parent can pass on is one big Y with one big R. You can write it as many times as you want, but in reality, you only need to write it once. Okay, so they must pass on one allele for each trait. If we look at the second parent, the only gamete that can pass on is a small y with a small r. So you'll just see me writing out, must pass on a y and an r together. You cannot just pass on y comma r. They must be passed on together. And then we do our cross, which is also known as our Punnett square. I always like to say that one parent's gametes go down along the side, the other goes along the top. Because we started with Y's the whole way along, we are going to continue keeping our Y's at the start. So a big Y with a small y gives me big Y small y, and a big R with a small r gives me big R small r. So that is the only combination of offspring that I can have. Remember, your offspring are known as your progeny, and F1 refers to first generation. So we just copy out the genotypes from the cross, big Y, big R, small r. The dominant is present, so this is heterozygous for both traits. We have a big and a small for both traits. But because the dominant is there, the dominant is going to be expressed. Big Y codes for yellow and big R codes for round. So all of my phenotype, my physical appearance of my offspring is going to be all yellow round. The dominant will always be expressed unless you were told in the question that there is incomplete dominance. So just watch me finish the rest of this and then we will go on and look at an incomplete dominance cross. Okay, so I know this video is longer than my usual ones, but I do actually want to give you time to read through it, so that's why it's going so slow. Now we have an incomplete dominance cross, and incomplete dominance means that neither allele is dominant nor recessive over the other. So if we have like a capital R and a small r, and we have the heterozygous big R, small r, usually it looks like the dominant parent. However, with incomplete dominance, no allele is dominant over another. So flower colour shows incomplete dominance, and this means that the heterozygous genotype, big R, small r, is pink, not red or white. Big R, so 2 big R's is red, and 2 small r's is white. Find the genotypes and phenotypes of the progeny produced by crossing a red flower and a white flower. So I always like to write a little note for myself that big R is red, small r is white, and big R, small r is pink. You only need to worry about this if you were told in your question that there's incomplete dominance or something is not completely dominant. Otherwise, proceed as normal that the capital letter is dominant unless you are told otherwise. Okay, so then the step that I like to do next is write out my parental phenotypes, what they look like. So I am told to cross a red flower with a white flower. So I just copy down those. Remember, pH for phenotype, pH for what they physically look like. So red flower crossed with a white flower. Then I do my genotypes. Remember, there's only one combination in incomplete dominance that will give me a red flower, and that is 2 big R's, homozygous. My only combination for white flowers is 2 small r's, also homozygous. Then I do my gametes. My first parent can pass on a big R, my second parent can pass on a small r. One parent's gametes go down the side, the other parent's gametes go along the top, and I cross them. A big R with a small r, I have big R, small r. That's the only offspring that I can produce, or progeny. So my F1 first generation offspring will all have the genotype big R, small r, which is pink. Because remember, heterozygous, we're told in the question, is going to give pink. You would not believe how many people get to the very end of this and write down red. Make sure you spot that incomplete dominance. They're telling you in the question that heterozygous is pink. They will not always spell it out for you that obviously, okay? So it is one to note and look out for. But they are my examples of a dihybrid and an incomplete dominance cross for genetic crosses. While genetic crosses has appeared majority of years in order for you to do, there has been 4 years where you haven't actually been asked to physically draw out your genetic cross. Theory is absolutely crucial because you need to interpret the questions that you've been given. Hopefully you found this useful. If you did, please like it, subscribe to the channel, comment below. My genetics booklet that I'm working from in this is available on my website leavingcertbiology.ie. I also have completely free podcast episodes and videos on social media, TikTok and Instagram, where I go through everything that you need to know. But for now, I'll talk to you soon.